Good evening, everyone, and welcome to Future's webinar series. I'm Anne-Marie webinar coordinator. I want to celebrate International Women and Girls Day in Science, remembering all those superwomen who wrote their names in history of science. I also use this chance to invite and encourage girls to choose this path. Cheers to that. Our guest tonight is wonderful science super lady, Dunja Aksentijević. She was born in Bosnia, and her education and career path took her to the most prestigious universities in the United Kingdom. She will tell us not just her life story, but also about her research in the field of cardiac metabolism, balance of energy in heart, and what she and her team find out may help treating heart diseases. Professor Dunja, it's an honor to have you.
Dobro večer, Ivana. Dobro večer svima vama i hvala na vašem uvodu. Ogromna mi je čast biti večeras sa vama u virtualnom barem diljem cijele Bosne i Hercegovine i veliki pozdrav iz smrznutog snježnog Londona večeras. Tema mojeg večerašnjeg izlaganja je naučna tema kojom se inače godinama bavim. Šta je to moć kardiovaskularnog metabolizma? Ono što nas hrani, nas na kraju ubije, kao što kaže latinska izreka. Ja sam trenutno vanredni profesor kardiovaskularne fiziologije i metabolizma na William Harvey institutu, Queen Mary univerziteta u Londonu ili Bartsove katedre za medicinu. Također sam fellow Britanske fondacije za srce i Wellcome Trusta, kao i počasni istražni član King's College u Londonu i počasni istražni član univerziteta u Oxfordu. Prije nego što prijeđemo na naučni dio moje prezentacije, zamolili ste me da vam u kratkim crtama objasnim moj put akademski u moje karijeri do sada. Kao što ste u uvodu već spomenuli, ja sam rođena u Bosni i Hercegovini, ja sam rođena u Tuzli i iz gimnazije Meša Selimović iz drugog razreda sam dobila George Soroševu školarinu nakon intervjua i selekcije. Dobila sam školarinu da 3. razred gimnazije provedem u britanskoj privatnoj školi, u ovom slučaju to je bio Queen's College u Tauntonu, jedna od prestižnih privatnih škola, što vidite na slici ovdje. Tako da sam zamijenila gimnazijsku klupu u gimnaziji Meša Selimović vrlo drugačijim svijetom britanskog školstva u kome sam zaista uživala. I nakon godinu dana, uspješnih godinu dana, dobila sam punu školarinu da završim svoje srednjoškolsko školovanje u Velikoj Britaniji, što je vodilo dalje u punoj školarini fakulteta za nauke na univerzitetu u Hullu, da mi dodijeli fond za moje studije, dodiplomske studije iz biomedicinskih nauka i genetike. Ovdje mi je bilo zadovoljstvo naći fotografiju kada sam sinoć pripremala ovu prezentaciju, jedan od mojih prvih eksperimenata koji sam radila u laboratoriji. U pitanju je bila ljetni program i školarna koji sam dobila od Wellcome Trusta, radim projekat sa tadašnjim mojom profesoricom, koja u stvari ovaj projekat ljetnji je pokrenuo moj interes za kardiovaskularni metabolizam i moj inicijalni naučni trening. Ova slika je tačno prije 19 godina iz laboratorije za kardiovaskularni metabolizam na univerzitetu u Hullu i u pitanju je bio eksperiment ćelijskog metabolizma u ishemiji i reperfuziji. Tako ova prilika je idealna da se vratim malo u te prve dane i prve korake moje naučne karijere koji su mi dali inspiraciju i snagu da dalje nastavim istraživačkim putem. Tako da nakon završenih sam radila istraživanje iz kardiovaskularne inzulinske rezistence u bolestima bubrega i odmah nakon završetka doktorskih studija, mislim doslovno do 7 dana nakon moje odbrane doktorata sam bila pozvana na intervju na univerzitetu u Oxfordu, gdje sam se pridružila kao naučni saradnik, postdoktoralni naučni saradnik grupi profesora Stefana Neubauera, jednog najeminentnijih imena iz kardiovaskularne energetike i metabolizma u svijetu. Tako da sam imala ogromnu čast, izazov da odmah poslije svojih doktorskih studija se pridružim jednoj tako dinamičnoj velikoj grupi na Oxfordu. Tako da sam na Oxfordu se zadržala narednih nekoliko godina kao istraživač, a također sam bila aktivno član i nastavnog procesa kao profesor i kao tutor za prijem na medicinu i na fiziologiju. Karijeru sam nastavila na King's College u Londonu kao viši naučni saradnik I poslije mojih 10 godina postdoktoralnog treninga sam dobila poziciju na fakultetu za prirodne nauke na Queen Mary univerzitetu u Londonu kao vanredni profesor fiziologije i hemije i sada sam profesor kardiovaskularne fiziologije i metabolizma na medicinskom fakultetu, odnosno William Harveyu. Tako da je to neki moj put od gimnazijskih klupa do večeras kada imam da zagovaram s vama i da podijelim neke od mojih naučnih rezultata koji sam ostvarila u zadnjih 10 godina. To bi bio dio uvod, tako reći, generalni o mom dosadašnjem radu, a sada bih ipak prešla na naučni dio moje prezentacije, koji ću nastaviti na engleskom, tako da ćemo sad promijeniti jezik. So for the last 10 years, or last 15 years, I have been interested in cardiac metabolism and heart failure, in the context of heart failure. Why? Because it is one of the biggest health challenges we're facing today. It's one of the biggest global health challenges that used to be reserved only for the developed nations. However, it has also become very much a problem of developing societies. And as you can see from the numbers that are presented by World Health Organization, we have death toll of about 17.5 million lives a year. And this disease, heart failure, affects over 64 million people. Therefore, it is before Corona and COVID became a headline as a pandemic. Realistically, prior to that, as a world, as a society, all societies are facing a huge heart disease or cardiovascular disease pandemic, claiming many lives and costing an awful lot economically to societies. Recently, my students at Queen Mary, as a part of their master's program, had a pleasure to hear a presentation by my ex-colleague from Oxford, who now works for Novo Nordisk as a scientist in heart failure research, Dr. Marie Seymour. And she was presenting some very elegant work that they're currently doing in trying to find novel therapies for heart failure. And she used this expression for heart failure to be a graveyard of clinical trials. So despite enormous public health need for drugs for heart failure to reduce the mortality, to improve the quality of life, is it safe to say that the scientific community and pharmaceutical industries really have so far failed to find any magic pills to fix the problems with the failing heart? So, can we find success amongst the wreckage of all the failed studies to try to find clinical trials to treat heart failure? So, where does this cardiac metabolism of interest of ours fit in? Well, failing heart, we have known for the most of 20th century, is something referred to as engine out of fuel. In the 1930s, German physiologists have actually done biopsy analysis, biochemical analysis of post-mortem cardiac samples, and have noticed that failing heart patients all have reduced creatine or reserved energy reserve. So this was the origin of the engine out of fuel hypothesis for the metabolism in a failing heart. So if we have known that this has been a problem physiologically, biochemically, medically for the last almost 100 years, still metabolism and function in the heart, we know, are linked. Very few investigations have actually considered energy substrate metabolism as a paradigm for treatment of heart failure. And as such, the energy metabolism as a target for development of new drugs by pharmaceutical industry has largely eluded the attention of pharma to try to tackle it as a target to treat heart failure. Again, why are we interested in this particular aspect of cardiac physiology? The heart is a very complex organ, and this complexity of myocardial anatomy, hemodynamics, and coronary flow may obscure the very simple fact that the heart is an incredibly efficient converter of energy. It is also an enormous user of energy. So in the series of highly enzyme catalyzed reactions, or highly regulated enzyme catalyzed reactions, heart muscle converts chemical energy into mechanical work that keeps us literally alive. It is the #1 ATP consumer in the body. it goes through 6 kilograms of ATP every day, or 20 to 30 times its own weight. And all of this to support the work of 100,000 beats, or pumping 10 tons of blood per day. And normally I say to my students during my lectures when I show this slide is, next time you go to the supermarket, grab a few water bottles and see how much 6 kilograms really feels. That is how much chemical energy our heart utilizes every single day, which is phenomenal. Given the size, it's an organ the size of probably manly a sized fist than mine. So what gives heart all this energy and all this ATP in essence? It's mitochondria. Heart is incredibly rich in mitochondria. About a third of cardiomyocyte volume is filled with mitochondria in order to meet this ATP, to synthesize this ATP and meet this ATP demand. If you quickly have a look on any chemical supply website, which we use on a regular basis in the cardiovascular research to buy our supplies for daily experiments, if you were to buy this ATP, it would cost you just for days worth of work. And this is just for a healthy heart. This is not a hypertrophied heart. This is not a heart in pregnancy. This is not a heart of athletes who may go through extreme bursts of energy demand. Just to meet this general daily energy expenditure, if you were to buy this compound from any chemical company that we get our ingredients for experiments from, you'll be looking at 300,000 pounds a day, or 600,000 convertible marks. So it is incredibly precious organ in every single way, every single sense. So in order to meet this ATP demand, obviously there's a very complex machinery metabolically that's behind the scenes to ensure that we can synthesize sufficient amount of ATP. However, as complex as it may be, realistically can be broken down in terms of regulation into very simple stages. So stage one is a fuel from food. This is substrate utilization or import of substrates from blood into the cardiomyocytes. The stage two is processing of external fuels to metabolic intermediates that feed into Krebs cycle and oxidative phosphorylation ultimately, which is the site of oxidative ATP synthesis. In addition to the steps for ATP synthesis, we also have energy communication pathways, which are crucial for this ATP matching of ATP supply and demand in the cell in the form of creatine kinase mediated enzymatic reactions, which link the site of ATP synthesis in mitochondria to the ATP sinks or sites of ATP utilization in the cell, such as SERCA and myosin ATPase. So alterations in these individual steps of cardiac metabolism I've just outlined for you, such as changes in substrate utilization, intermediary metabolism, and also changes in high energy phosphate metabolism, all lead to that energy starvation or engine out of fuel hypothesis from the beginning of my talk that we see in heart failure. So in the normal healthy heart, we have a defined metabolic phenotype that's dominated by utilization of fatty acids as a main source of ATP over glucose. Heart is an omnivore, so it can utilize a whole host of substrates, carbohydrates and ketones and amino acids in some circumstances, but really adult healthy heart does have this metabolic hallmark of using fatty acids to support its ATP provision for work over glucose. And the reason is very simple. It comes down to the actual ATP yield that we get after breaking down fatty acids per every oxygen molecule consumed. In a healthy heart, with this type of substrate phenotype, we also have very healthy energy reserves. So this is the phosphocreatine to ATP ratio in cardiomyocyte. So what I'm showing here on the slide is just an example of a phosphorus spectrum, Phosphorus Nmr Spectroscopy. This particular example has been taken from a perfused rat heart in one of my previous studies. And here we can see a very healthy ratio of phosphocreatine as energy reserve to 3 peaks of ATP. And we also see a phosphate peak here as a result of the breakdown. So a healthy adult heart or healthy heart has a healthy energy reserve. I've seen here a PCR to ATP ratio, which is the definition of energy reserve, is comparable between regardless of which heart you look at. And this is a result of this healthy substrate utilization. What happens in diseased heart is in a hypertrophied heart, in a failing heart, is we have a switch from this adult metabolic phenotype characterized by fatty acid use to enhanced glucose utilization, which may not be problematic initially. However, long-term wise, glucose is simply not ATP rich enough to support long-term work. Therefore, this switch from fatty acids to glucose is something we define switch from adult to fetal metabolic phenotype. Why? Because it's during the fetal developmental stage that we rely heavily on glucose use for ATP provision over fatty acids. And unfortunately, the long-term switch in substrate preference from fatty acids to glucose leads to impairment of energy reserve in heart. So what you have here at the bottom of my slide is example of phosphocreatine phosphorus spectroscopy, again acquired from beating hearts, where you see a difference in the peaks of phosphocreatine between healthy and diseased heart, and changes also in resolution of ATP peaks, which are broader and smaller. And you have, drop in PCR to ATP ratio as a result of this long-term change in metabolism, metabolic profile, and metabolic remodeling. When I mentioned to you I had a huge honor to go and work with Professor Noba in Oxford, he has really defined or contributed significantly to awareness of importance of cardiac energy reserve in a healthy and failing heart, because his seminal studies from 1990s have shown that if you take this actually spectra, phosphorus spectra obtained from humans, from patients, from healthy volunteer, and from dilated cardiomyopathy patients, is that in comparison to a healthy volunteer, we have nicely resolved PCR to ATP. At various different stages of dilated cardiomyopathy, this peaks, as you can appreciate, even for those with non-medical background, are disappearing, with the spectrum shown at the top of the patient that was dead within 7 days of MR. And this drop in PCR to ATP ratio in all the studies done has become one of the strongest predictor of cardiac mortality in comparison to any other clinical parameter. So if patients present to clinics with a very poor PCR to ATP ratio, unfortunately, it is time to act very quickly, or the prognosis is not very good. So in terms of during my doctoral studies, I looked at the chronic kidney disease model or cardiomyopathy, because unfortunately renal failure patients usually suffer from cardiomyopathy with lethal outcome. We've shown that this is a classic example of remodeling, where you start with cardiac hypertrophy, change in substrate utilization, and changes in insulin sensitivity in terms of response of heart to the external metabolites, drop in PCR to ATP ratio. So you start with gradual pathophysiological remodeling, substrate switch, change in insulin sensitivity, leads to energetic compromise, and overall, if left untreated, leads to death. In addition to heart failure just alone, another pandemic we are facing is also the type 2 diabetes, with millions of people around the world being affected, and again, both developing and developed countries. So again, type 2 diabetes is characterized by enhanced risk from cardiovascular disease, specifically from diabetic cardiomyopathy, affects over 500 million people worldwide. And everyone with type 2 diabetes has enhanced risk of dying from heart disease. And also, part of my current research at Queen Mary, I'm looking at how inflammation is a part of that story. And whilst I've shown you that remodeled heart in hypertrophy and failure switches from fatty acids to glucose, the problem in diabetic heart is that we have absolute reliance on fatty acids to the point where it's actually killing the heart, because too much fatty acid use, a very fatty heart, leads to energetic deficit equally as a heart that can't utilize any fatty acids, purely because heart needs that seesaw energetic flexibility to switch between different substrates in order to meet varying ATP demand. So this high reliance on fatty acids in the milieu of metabolic excess, such as hyperglycemia and hyperlipidemia, which we have in type 2 diabetes, leads overall still to energetic deficit. As it progresses, we get reduction in PCR to ATP ratio. And this is again part of my current work looking at type 2 diabetes. You get usually accompanied by obesity and very fatty hearts, hyperglycemia, so liver phenotype, enlarged hypertrophied heart that ultimately in type 2 diabetes lead to heavily remodeled and energetically impaired heart. But sometimes we also have to look past. When we think about disease, a disease such as heart failure, when we look at diabetes, we also may have to look at and normally, obviously, our laboratory animals and models that we use in our research are rats and mice, and past also sometimes guinea pigs and rabbits. But sometimes if we try to think of novel heart failure drugs, perhaps it's also time to look at the nature and look at the organisms that have adapted quite well to kind of metabolic stresses and conditions that are almost lethal for us. So my current collaboration is with Professor Stephen Rossiter from Queen Mary School of Biological and Chemical Sciences, where in fact we're looking at the adaptations in sugar-eating bats that their diet almost exclusively relies on glucose and sucrose and carbohydrate loads, yet they never develop type 2 diabetes. They never have problems that we see in diabetic heart. Therefore, sometimes we have to look back to nature or organisms that have adapted to extreme metabolic stress states in order to give us an idea how to tackle our own disease process. Equally, if we look at a metabolic phenomena back again in nature, and this is in collaboration with Dr. Chris Fawkes at Queen Mary University of London, if we look at a naked mole rat, this beautiful looking chap here, in comparison to our normal lab mouse, which is, you know, in essence, an average Joe like us, naked mole rat can survive almost 20 minutes of complete hypoxia, so no oxygen. If we starve our heart, even transiently, of oxygen, we're going to have irreversible damage, both physical damage to the muscle, metabolic damage. Yet naked mole rat can survive on regular basis throughout its life, which compared to a normal mouse that lives 12 or 24 months, naked mole rat lives 30 years, has adapted to survive these extreme challenges, such as hypoxic challenges, which is equivalent of us having heart attacks and strokes. And when we looked at the metabolism of these hearts, we have noticed that they have remodeled their metabolism, their normal metabolism, for example, to have a huge glycogen reserve in their heart they can tap into potentially when they have energetic stress and shortage of oxygen for normal oxidative metabolism. It is easy to accuse the heart of just giving up when there's a challenge and just switching metabolism and going into energetic deficit. But equally, my work over the years has also provided examples of extreme energetic plasticity or adaptability. And it's amazing kind of insults we can throw at the heart and still see extensive adaptations in order to keep going, because quite frankly, it's not an organ that can stop, unload, rethink and start again, because we know if it stops, it's a game over. So one of the studies I've done when I was in Oxford, Professor Neubauer, is looking at which other systems step in when we have a failure of the major energy communication pathways, these wires that connect energy supply and demand. And that was in the form of glycogen. We discovered that when we have changes in creatine kinase activity, which is the main modulator of phosphorus transfer in the heart, that's the fifth component that I was showing you of metabolism, we actually have few enzymatic systems that try to step in to save the day. So if we have a reduction in creatine kinase, we have a upregulation of different enzymes called adenylate kinase to mediate phosphorus transfer. And also, it's almost call a friend situation when some of the enzymes of glycolysis start doubling up also as phosphorus transfer enzymes. And we have seen this in different models of heart failure, whether it was a post-myocardial infarction model, or whether it's coronary artery ligation model or high pressure overload model. So regardless how we induced heart failure, we saw this myocardial attempt to, in essence, compensate for impairment of the main creatine kinase mediated phosphorus transfer. Fortunately, bad news is in long term, if you strip, for example, creatine from the heart, which is a principal component of your PCR, phosphocreatine to ATP energy reserve, that yes, you can have short-term adaptations, like some of them I've just shown you, but unfortunately, long-term effect is going to lead to cardiac dysfunction and also going to lead to extensive mitochondrial damage and remodeling, and change in organization, change of reactive oxygen species production, etc. So yes, our heart is very plastic, or it has a level of adaptability. However, if you long-term strip out key components of energetics in heart, you're going to have quite a detrimental effect. And there's also cautionary tale of playing metabolism in the other end, so not deleting things or removing things. But equally, if we had too much creatine in the heart, so trying to provide too much of energy replenishment to myocardium, the result was still failure and hypertrophy, because it's this fine balance that's maintained in myocardium that keeps it going and keeps it sufficiently supplied with ATP. Equally, sadly, we have another story of where extreme modulation of metabolism in attempt to find a new metabolic cure for heart failure backfires. Because, for example, when we try to target the portion of fatty acid metabolism, specifically malonyl-CoA carboxylase, we found that actually transgenically deleting this component of metabolism, which we thought is going to be very exciting because it's potential a drug for, because it will cause a shift to glucose oxidation and therefore its therapeutic potential for myocardial ischemic injury, for example. We found that these mice were just dead because they couldn't even survive past the postnatal stage, because simply this inflexibility of metabolism in completely removing fatty acid oxidation from the story has not been helpful at all. They developed a heart remodeling or impaired cardiac function, cardiac hypertrophy, and they had actually depleted ATP or total adenine nucleotide pools. So by trying to modulate metabolism in hope to find a new drug, we have also seen how it can easily go to completely opposite way, either by deletion or overexpression. So therefore, the first conclusion of my talk is that I hope I've convinced you that maladaptive metabolic remodeling takes multiple forms across multiple energy handling domains, and that lack of metabolic flexibility in reserve in terms of substrate and utilization, oxidative metabolism, and phosphotransfer represents a common denominator, ultimately which compromise energetic efficiency and contractile reserve, and that we really need a better understanding. I hope some of failure stories I presented to you demonstrate how we better understanding of the cardiac metabolic phenotype and metabolic signaling in heart failure, and how we go from pathological hypertrophy to the failing heart. The second portion of my talk, I'd like to actually focus on my recently published work, which was really result of many years of hard work that I have done since coming from Oxford to King's College, where I have looked at the impact of intracellular iron homeostasis, specifically sodium, on cardiac metabolism. And this is the work that was recently published in Nature Communications in just August last year, and it actually garnered a lot of media attention in terms of, because in essence we have identified novel mechanism in heart failure that's could be potentially targeted with novel drugs, and also has been subject to press releases by both King's College and Queen Mary, which as a scientist is a huge honor, because when your home institutions highlight your own work in an environment where there's no shortage of excellent internationally reputable scientists. So why sodium? Well, cardiac hypertrophy and failure, in addition to metabolic remodeling, which I've just shown you, is also characterized by sodium overload. So I apologize for a very small print. This is actually a table taken from one of our reviews. From mouse to man, we have changes in sodium in hypertrophy and failure. And this is seen, like I say, across species, different species, regardless which technique used to measure sodium. Definitive hallmark of pathologically remodeled heart is sodium overload. We have also seen this in human and animal models of heart failure, and also it is very clearly seen just from acquiring some nuclear magnetic resonance sodium spectroscopy from the heart. So you can see changes in intracellular sodium from a healthy to hypertrophied heart. Sodium homeostasis actually has been targeted therapeutically for the past 200 years, because obviously people at the time didn't know the ins and outs of intracellular sodium regulation and intricacies of molecular regulation. However, the drug digoxin, that's been around for 200 years and has been used for treatment of heart failure, is in fact a sodium potassium or sodium pump inhibitor. But we know now it's not the best drug, it's a very poor drug, and most certainly our study from King's that's been recently published has shown highlighted why it should not be used for treatment of heart failure, because it causes a huge energetic stress on the failing heart. So what causes this rise in sodium that I've just introduced to you? Well, cardiac sodium pump, or sodium potassium ATPase, plays a crucial role in transmembrane transport, iron homeostasis, electrical excitability, control of cell volume, and contractility. So there are many ways in which sodium can enter cardiomyocyte. There's only one principal extrusion pathway, or the route via the sodium potassium ATPase. And the activity of sodium pump, or sodium potassium ATPase, is regulated by a protein called phospholemon. Phospholemon is a member of fixed family of proteins. It gets phosphorylated and dephosphorylated by PK and PKC, therefore is the gatekeeper of sodium pump activity. However, it is not very clean cut what causes this sodium overload, because the jury is still out whether it's increased influx or decreased efflux that causes the sodium overload that we see in hypertrophy and failure. Most certainly work from my colleague from the lab, Andriy Boguslavsky, has shown that in transgenic model of sodium pump chronic inhibition, which is a phospholemon 3SA mouse, we also have increased susceptibility to hypertrophy-induced dysfunction, where the pump is, in essence, chronically impaired. Also, another question that's been in literature is what comes first, the sodium elevation or metabolic remodeling, so chicken or the egg question. And if you look at the name of the pump itself, sodium potassium ATPase, it indicates obviously the pump is a ATP utilizer, and in fact a significant one in the cell in terms of it's a chunk of ATP it takes from all the ATP that gets synthesized. So one of the ways that people tried to build this relationship in the past between a sodium elevation and shortage of energy in the cell was simply saying, well, maybe pump doesn't get enough ATP because of the metabolic remodeling and substrate switch. This inhibits the pump and therefore sodium accumulates. However, that's not necessarily been shown to be the case, because both delta G for ATP and concentration of ATP even at lowest don't inhibit the pump. And this was even seen in low flow ischemia studies where the flow was minuscule to nothing to the heart, yet the sodium pump still had enough ATP to function. So we have actually focused on the work that's in the early 90s by scientists called Cox and Matleb, who have shown or suggested from the work in isolated mitochondria and isolated cardiomyocyte, it's perhaps the rise in cytosolic sodium that drives the activation of mitochondrial sodium-calcium exchange, which would cause calcium to efflux from mitochondria, therefore having an impact on ATP synthesis. Why? Because mitochondrial calcium is a key regulator of key mitochondrial dehydrogenases that are responsible, in essence, the key steps, regulatory steps for ATP synthesis. Therefore, changing the calcium availability mitochondrial matrix by activation via high sodium could potentially impact the ability to synthesize ATP mitochondria. Therefore, they have made a suggestion or made this hypothesis back in the early 90s, but the extent to which these metabolic changes reflect chronic cellular remodeling or arise as a consequence of sodium elevation have not been determined. So we decided to ask a question. Is there a causal link between acute and chronic sodium overload and metabolic remodeling in the heart? And our hypothesis was that metabolic dysfunction, the mismatch in ATP supply and demand, is secondary to sodium-potassium dysregulation and intracellular sodium accumulation. And ultimately, we asked the question, does pathological hypertrophy share a common sodium-mediated metabolic fingerprint with acute and chronic sodium overload? So we have used integrated experimental approach. So my background, my training in cardiac physiology and metabolism includes a lot of heart perfusions, ex vivo heart perfusions. This is an example of a heart perfusion laboratory at St. Thomas's Hospital at King's College London, where I have worked for years. So this is what we normally use for our perfused heart Langendorf experiments. However, we had additional challenge. We want to study energetics and sodium homeostasis using NMR spectroscopy, which is also part of my expertise. So we had to build a system called umbilical perfusion rig, shown here. So we have changed what you see here in terms of the glassware on the bench to a umbilical type rig that's completely made of NMR compatible parts, non-metallic, that can fit inside the magnet and keep the heart alive whilst we acquire a series of biochemical spectra. So this is an example of experiment. This is a mouse heart being perfused under physiological temperature, oxygenation, the buffer containing nutrients. It's being dropped into the bore of 9.4 Tesla magnet, where it's going to continue to beat. And we're going to acquire information in terms of sodium homeostasis, energetics using sodium and phosphorus spectroscopy on beat-to-beat basis. This is an example of the mouse heart beating in an NMR tube. And we have also designed a system so we can very quickly switch between solutions, so we can study impact of different drugs. So there's no issue in terms of visualizing or quantifying sodium in heart, sodium, because in essence there's plenty of it. But the problem is, how do you separate what is extracellular sodium and what is intracellular sodium? Because sodium outside of the cell is going to be in the order of magnitude of 148 millimolar. The intracellular sodium is about 8 millimolar. So as you can see from the in vivo sodium NMR imaging that's been done actually recently in collaboration with the colleagues at the University of Aarhus in Denmark, what you see brightly shining in the image is a heart, and you can see this is a sodium image of the heart. So there's plenty of sodium. But if you try to visualize how much of that is intracellular versus extracellular, this tiny little shoulder on top of the peak is your 8 millimolar sodium in the face of 145 millimolar extracellular sodium. So traditionally what we have used is something called shift reagents, which are chelators of cations, to perfuse the hearts, shift this extracellular peak from intracellular peak in order to quantify how much intracellular sodium we have in heart. The problem with this approach is that chelators also chelate the calcium. Therefore, the moment you chelate all the sodium to visualize sodium, you also chelate calcium, you end up killing off cardiac function, which, as you would agree, not exactly an ideal approach if you have to study beating metabolism in a beating heart. Therefore, with the help of a colleague from King's College London, Dr. Thomas Ekin, who has previously worked on sodium spectroscopy in red blood cells, we have established a non-invasive, not relying on any agents or shift reagents, technique for quantifying intracellular sodium, multiple quantum filtered sodium spectroscopy. What we have done, in essence, is used the behavior of sodium in the cell to separate the signals from extracellular to intracellular sodium, and we have gone through a series of experiments to validate this approach with the various sodium concentrations, using drugs, using modified buffer composition, in order to validate this technique prior to applying to our studies. And also by selective digestion in the heart using saponin and Triton, we have also managed to analyze what portion of this total sodium signal we get from the heart comes from cytosol or comes from mitochondria, which is a site of interest for our hypothesis. So therefore we have used integrated experimental approach, combining physiological techniques with NMR spectroscopy, such as phosphorus spectroscopy, and I've already shown you the phosphorus spectra, so this example. We have established high resolution proton metabolomic profiling for looking at the metabolomic profile of the hearts, established carbon-13 substrate preference and flux assessment, and also used mass spectrometry to supplement our biochemical analysis. So, in terms of a chronic sodium overload, we have used two models. We have used transgenic mouse model and more physiologically relevant cardiac hypertrophy model. So you can see here what hypertrophied hearts looked like at the end of a 5 weeks post trans-aortic constriction surgery. And what we found in both of our chronic sodium elevation models, that indeed we did have sodium elevation, and it was to similar extent between both transgenic model of sodium pump inhibition and the model of cardiac remodeling, LVH. So first we embarked on a series of studies to look at the impact of chronic sodium elevation and metabolic modeling. So we have performed our NMR studies as described, I was just showing you a second ago. So we have acquiring sodium and phosphorus spectra from perfused mouse hearts, and also repeated all these studies under more metabolically physiological conditions, in essence, by including all the metabolic substrates you normally get in the blood in perfusate, to make sure that we are not limiting our hearts only to just glucose in the buffer, to make sure it's physiologically more relevant. What we have found is that chronic sodium elevation did not impair the PCR to ATP ratio, did not impair energetics. And this was seen in both a transgenic model and also at the stage of hypertrophy where we examined the hearts. So we did not have so high sodium chronically at this stage we examined did not cause changes in energy reserve. However, what we did find that this maintenance of cardiac energetics did still come at metabolic cost, because the hearts had chronically changed substrate preference from fatty acids to glucose. And this was seen in both models of sodium elevation, both transgenic model and hypertrophy model. Therefore, it was independent of etiology of elevation. We had similar profile of substrate switch. So we have known before the substrate switch from fatty acids to glucose is very much a hallmark of the failing hypertrophy heart, but this has never been shown before to be triggered, in essence, by sodium. In keeping with our hypothesis, we used the inhibitor of mitochondrial sodium-calcium exchange, CGP 137157. So we have repeated all the experiments with this inhibitor present acutely, and we were astonished to find that even with the acute inhibition of sodium-calcium, mitochondrial sodium-calcium exchange, we rescued the substrate switch from this pathological fetal phenotype-like switch from fatty acids to glucose back to being normal again, in fatty acids being the main source of ATP. It was actually quite incredible that this switch was so immediate. We also found that in addition to substrate switch, that chronic sodium overload again comes at a cost, or maintenance of cardiac energy reserves come at a cost, because multiple components of energy, intermediate energy metabolism, were depleted. And again, with treatment with CGP, we managed to rescue these levels, rescue level of metabolites, both in our transgenic model and our hypertrophy model. In terms of acute sodium overload, and this example of that, for example, is heart attacks. When we have ischemia, we have immediate rise in intracellular sodium. So we have induced acute sodium overload using pharmacologically, using Warbane, the sodium pump inhibitor, and we titrated that Warbane with the blebistatin, which is to have a mild uncoupling effect. Why? Because we didn't want to produce any positive inotropy in our hearts when we inhibited the pump, which would normally happen. And therefore, we want to make sure that acutely we had elevation in sodium without any changes in the cardiac inotropy causing extra demand on cardiac energetics. We want to see just what happens between sodium and metabolism. So we repeated our study. This is a protocol, a calibration of perfused hearts, and then perfusion with the buffer containing Warbane and blebistatin. We did again our sodium phosphorus proton spectroscopy, and again repeated that with all metabolic buffers on the bench. And again, similar to our chronic, first of all, we found that this is a protocol that despite immediate rise in sodium when we inhibited the pump, we had no increase in left ventricular developed pressure. Therefore, the hearts didn't have extra demand in terms of function. As you can see here, in absence of blebistatin, your LVDP goes up immediately when we inhibit the pump. Yet, our Warbane blebistatin mixture absolutely did not change left ventricular developed pressure and energy demand in the heart. Yet, the sodium was high. And what we found is that PCR to ATP ratio, again, in this short period was not inhibited. We're talking about half an hour sodium pump inhibition, so acutely. But we had a similar profile of substrate switch from fatty acids to carbohydrates. And in the case of acute sodium stress or acute sodium overload, the actual extent of metabolic depletion was much more severe. So keeping the energetics going, keeping the PCR to ATP ratio definitely came at the cost of substrate depletion. And when we repeated the acute sodium overload studies with mitochondrial sodium-calcium exchange inhibitor, we found that this again reversed the metabolic remodeling. So all the depletions that we saw in intermediate metabolites were rescued by the CGP incubation. However, we're not happy to stop there. We wanted to use all experimental data to try to build even bigger picture to what happens to metabolism with sodium overload. So we have collaborated with colleagues in America, in Texas, the legendary cardiac metabolism scientist, original Krebs student from Oxford, Dr. Heinrich Tegmayr, and Anja Karlstadt, my colleague from her early training days in Berlin. And we have used Anja's amazing in silico model of human cardiomyocyte, which is CardioNet, to try to model over 1,700 metabolic reactions across different metabolic compartments. And we found very interesting that we found some common metabolic changes in the presence of high sodium. So if you look at our control hearts, this is principal component analysis of metabolism, control our hearts are here, and all our sodium acute and chronic groups are completely overlapping. So regardless how long the heart is exposed to high sodium, what causes it, metabolic profile is definitely similar. And again, looking at the flux balance analysis, we found a whole host of common fingerprint changes in metabolism, namely reduction in fatty acids, enhancement of carbohydrate metabolic utilization. And again, looking at individual fluxes, we found, again, enhanced metabolism of pyruvate, lactate, and amino acids, reduction in fatty acids, in acute sodium elevation, and in chronic sodium elevation, again, reduction in fatty acid oxidation, enhanced glucose utilization, and ketone body metabolism. Therefore, we managed to, there is definitely a metabolic fingerprint to this sodium elevation, regardless of duration and etiology. Therefore, in conclusion, a sodium overload results in common metabolic perturbations preceding energetic impairment. And this is regardless if it's acute, transgenically chronic, or chronic by hypertrophy, or metabolic, hypertrophic remodeling. We have substrate switch, reduced palmitate, and increased glucose oxidation, change in metabolomic profile in terms of TCA cycle, glycolysis, amino acid metabolism, however, without energetic impairment. And we found inhibition of sodium calcium exchanger by antagonist during both acute and chronic overload ameliorates these metabolic changes. And again, alit modeling has corroborated and further expanded our view in terms of what happens in substrate switch in these hearts. And therefore, the changes we observe definitely constitute a metabolic hallmark of sodium overload independent of etiology or duration. Collectively, we believe that these observations suggest that early prevention of sodium elevation could either prevent the origin or alter the course of metabolic derangement in pathological hypertrophy. So that would be so very quick summary of the work that we have done. Like I mentioned in the it's fully outlined in the paper that I mentioned at the beginning of my second half of the talk. And really, I would like to use this opportunity to acknowledge my colleagues who have worked with me tirelessly to put the story together. This team was led, the team and the study were led by Professor Michael Shattuck at King's College London, a very famous British physiologist who has been absolute inspiration and kind of person that every time you speak to, you learn something new. He's a walking physiology encyclopedia. My colleague, Dr. Thomas Ekin, who has helped us with sodium spectroscopy and validation of a technique, as well as my members of our Shattuck team, who have been amazing colleagues and supportive colleagues, as well as their physical input into the studies and various aspects of experiments. I would like to also acknowledge Professor Stefan Neubauer, who I worked with for years in Oxford, as well as my colleagues from Oxford, from both his group and other groups. My colleagues currently at Queen Mary, University of London, including Professor Frederica Merli-Berg, who I am currently developing a lot of new exciting projects. My long-standing collaborators from University of Cambridge. Tonight I didn't have a chance to speak about that aspect of our metabolic work and the succinate work we've been doing for years. Collaborators from US, collaborators from Birmingham, from Scotland, and many, many more couldn't fit really on this slide. And most importantly, all the various funding bodies supporting this work, from British Heart Foundation, Queen Mary, Faculty of Science, Diabetes UK, Barts Charity, AstraZeneca, as well as the Wellcome Trust, who recently awarded me my Wellcome Trust Fellowship. So I think this is the last slide of my presentation. So shall I stop sharing now my screen? Where I'm happy to take any questions.
Okay, thank you so much. This was really amazing and definitely very useful. It's inspiring to see people coming from our country doing important and wonderful things such as yourself. And thank you so much for being with us tonight. I'm sure you inspired everyone, especially young women, considering that today is the Day of the Women in Science. So I invite everyone to ask questions. If you want to talk to yourself, you can raise hand and I will allow you to speak, or you can ask it in the Q&A if you don't want to speak.
Thank you very much. I don't see any questions in the Q&A.
Yeah, and no one is raising hands, so don't be shy to ask any question. So As a researcher and a doctor, what do you recommend for a healthy heart? We all heard those advices, you know, do sports, eat healthy, but is there any specific advice that you can give to us?
It would definitely be advice in terms of change of our lifestyle habits. Once upon a time, smoking used to be a big culprit. These days, well, at least in UK, I suppose it's different in different places, and I'm not sure what statistics are for Bosnia. It's classic risk factors such as smoking, lack of activity. I mean, obesity is the #1 issue we are facing now. So the pandemic in terms of type 2 diabetes, one of the key drivers between heart failure problems that we see and cardiac disease is obesity and lack of activity. So there's a saying that sitting is the new smoking. So it's this sedentary lifestyle, very unhealthy diet in terms of high fat content, low fiber content. So switching over to Mediterranean diet, as they call it, which is high in olive oil and fish and vegetables and fruit, rather than processed, heavy, sugar-rich foods, really is the best way. It's the best start. Obviously, there are genetic factors and factors that we may not even be aware of in terms of what predisposes us to cardiovascular disease. But it's safe to say that if we have a healthy start and healthy lifestyle, we are going to definitely giving ourselves a better start or better outlook, because there's no drug that we can physically come up with in labs that can replace a fixed problem once actually a problem arises. Luckily, medicine has moved in leaps and bounds in terms of diagnostics, in terms of medical procedures, in terms of obviously therapy we currently have, but there's no magic pill to fix the problem once it starts. So the best intervention or best treatment strategy is prevention, really.
Thank you so much for the answer. Of course, especially with this pandemic situation and these stay-at-home situations, we need to even more take care of it.
Absolutely. No, COVID unfortunately has again highlighted all the ugly truths in terms of metabolic syndrome, in terms of diabetes, in terms of heart disease, because we have seen even in the short period, it seems like forever that we've been affected by pandemic. However, it is a fairly short period in terms of medical research and medical conclusions and findings. And it is very clear that the virus does affect, there's a worse outcome for those who are underlying cardiovascular conditions, hypertension, obesity, type 2 diabetes. So it seems that the pandemic that was there prior to virus arriving definitely predisposes patients to worse COVID outcomes. And a lot of work is currently undergoing to understand why this is the case. Obviously, a lot of work has been done in terms of ACE, the signaling via the ACE receptors, etc. So it's still still early days. However, the data or conclusions seem to be very much in line across the world and across. If you have an obese, diabetic, heart failure patient and you add COVID on top of that, it's not exactly a good mix. Yeah. So even the viral pandemic like this one has highlighted the importance of investment in cardiovascular health, really, as societies and individuals.
Thank you so much for that answer. We have one comment. I don't have any questions. I just wanted to say a massive thank you. I'm realizing now that physiology was and still is my favorite model. Really, really interesting.
Thank you. That's very kind. That sounds like one of my students. Thank you. That's a very, very kind comment. Thank you. Any other questions?
I don't see it.
Ah, it is one of my students. I was in year 1 last year. Well, thank you. Thank you very much. Thank you for attending, and thank you so much for your kind comment.
I will invite everyone to ask one more time, and if there is no questions, I would call on it.
Pitati pitanje na našem jeziku, mogu im odgovoriti.
Ok, znači ako niste sigurni na engleskom, možete pisati na našem jeziku. Tako da...
Ja mislim da bi to bilo vjerojatno to.
Ja mislim da ne treba više da čekamo.
Ukoliko neko ima, moji detalji su dostupni, ukoliko neko ima bilo kakvo pitanje ili želi stupiti kontakt, imate moju email adresu, imate, mislim danas su sve mogući profili dostupni online, LinkedIn profila, mog profila na univerzitetu, na stranici univerziteta, prema tome, ukoliko neko ima neka pitanja ili želi stupiti u kontakt, slobodno mi se javite, bilo mi zaista zadovoljstvo.
Hvala vam puno na ovom webinaru, na učešću, hvala vam na podršci koju ste pružili našoj fondaciji, kao i studentima u Bosni i Hercegovini, jer nemamo baš puno prilika da čujemo profesore svjetske klase poput vas, koji imaju ovakav ogroman uspjeh, ću reći. I stvarno osjećam se i ponosno i zahvalno zbog toga što ste iz naše države. Nadam se da ćemo se opet družiti ili u ovakvom nekom vidu ili možda čak i uživo ako nam to pandemija dozvoli. Vjerujemo vama istraživačima i doktorima da ćete što prije ovu situaciju riješiti.
Hvala vam puno na pozivu, zaista sam počastovana da sam imala priliku da barem virtualno budem dio podržim rad vaše fondacije i da se umrežim, tako reći, sa studentima u Bosni i Hercegovini. Ponavljam da sam zaista otvorena za vid svaki, ako mogu biti od pomoći ili saradnje, ako neko ima pitanje u vezi studija i ono o čemu smo večeras pričali, pošaljite mi e-mail.